Point mutations occur when one nucleotide base is substituted for another. This can have a variety of effects on the protein eventually produced by the DNA. Point mutations can occur via transition or transversion. Transition is when a purine base is substituted for another purine base, or a pyrimidine base is substituted for another pyrimidine. Transversion is when a purine is substituted for a pyrimidine, or a pyrimidine is substituted for a purine. This non-mutated DNA coding strand can be translated into mRNA and then amino acids that will form a protein. But what if some type of point mutation was to occur? How could this affect the amino acids that are produced by the DNA? There are several different types of point mutations that affect the amino acid sequence in various ways. A missense mutation is when substituting one base for another in the DNA sequence changes one of the amino acids that is produced. This may alter the protein, or could be neutral and not affect protein function. On the left is the original DNA sequence, and on the right is the mutated DNA sequence. Comparing the two, you can see that there has been a transversion, as a C base has been switched for an A. This will be translated into the mRNA and then into the amino acid sequence. In the original strand, asparagine was produced, but in the missense mutation strand, lysine was produced. A nonsense mutation is when a codon coding for an amino acid is changed to a stop codon. Comparing the mutated DNA sequence to the original, you can see that a transition occurred as a G base was substituted for an A. This translates through the mRNA and leads to what was a tryptophan amino acid being changed into a stop codon, so the protein would be cut short. A silent mutation is when the mutated DNA still codes for the same amino acid as the original. In this case, a transversion occurred as a T was substituted for a G. After translation into mRNA and then into amino acids, you can see that although there was a base substitution, the resulting codon still codes for alanine, so the mutation had no effect on the protein. A readthrough mutation is when a stop codon is changed to an amino acid codon, which results in a longer protein. Here, a transition occurred as an A was substituted for a G. This is translated into the mRNA and results in what was a stop codon in the original amino acid sequence becoming a tryptophan, and allowing more amino acids to be produced after it.